make a little organizational thing here. So the simple epithelia, you'll recall, I'm going to use a definition. I'm going to change the definition up a little bit. And that is, instead of saying it's just a single cell layer, which was, the good way to start the topic yesterday, I'm now going to define it this way and say a simple epithelium is one in which each cell contacts the basement membrane. Now that's going to be significant because when we start talking about a stratified epithelium, what we're going to find is that you're going to see clearly that there are cells which cannot possibly touch the basement membrane. But we're not there yet. So the definition is what did I write? Each contact, I'm glad I looked at that. Okay, each cell contacts the basement membrane. If I start acting erratically, somebody point this out, all right, and I'll try to moderate. All right, so we changed that definition up a little bit. Now, as far as the types, you'll remember, 3 of them have the word simple in them. Simple squamous, simple cuboidal, and simple columnar. All right, so that's just based on, like we said yesterday, the shape of the cell. And so if it's long and flat, it's squamous. If it's a cube, it's cuboidal. If it's rectangular, it's columnar. All right, and then we ended with at least showing you an image of this. And the last one is called pseudostratified. And I know that this engendered some concern because it's like, well, how do I know that's a simple epithelium? So, for example, this image right here, that epithelium is a simple epithelium. So you guys were looking at this saying, well, again, how could I possibly know that the last thing that looks like is a simple epithelium? So let me put your anxiety to rest by saying that the way you're going to know that it's a simple epithelium is by first recognizing that it's pseudostratified, and then secondly, knowing that pseudostratified is one of the simple epithelium. So you can actually work the opposite direction. So the key is to identify it as pseudostratified, and then we go from there. All right, and it's not going to be as bad as you think it is, or I hope it isn't. All right, so what I want to do is take a moment and illustrate for you what you're actually looking at, and this hopefully will help. Now, up until now, we haven't really mentioned too many cell names, have we? We have. In other words, with simple squamous, I showed you 2 specialized examples called endothelium and mesothelium. And I said that those would be considered as endothelial cells or mesothelial cells. But for the rest of it, I didn't really name cells. Now we're going to name cells. This becomes important. So here's pseudostratified. Let's start down here. Here's our basement membrane. Let's start with that. So this is the basement membrane. And so the lumen's going to be at the top of the picture. Now, the majority of cells are these sort of rectangular guys. And they have these extensions, these little hair-like extensions into the lumen, which happen to be cilia. And so these cells are called ciliated cells. I'll show them to you in a second. So that's an important observation. It's not the only place in the body where ciliated cells are found, but again, bear with me, we'll get there. But right next to a ciliated cell, there might be another cell that has this sort of interesting look to it, where it's called a goblet cell. So this thing is called a goblet cell because to somebody, I guess it looked like a wine glass. And sitting in here is actually a mucus drop. Now, let me just digress for a moment and say this. There is a molecule called mucin, okay? And mucin is, I don't know, it's a polysaccharide with some other stuff. I've honestly forgotten exactly what it is. But mucin, when it goes into solution, becomes mucus. Okay, you know what mucus is. Mucus, by the way, is a noun. It's a substance. But you're going to see an adjective oftentimes called mucus. Like some cell is a mucus this or a mucus that. So, well, there are no end to this, the failures here in here. So, the microphone. All right, so let's bravely push on. So, I mentioned, I think yesterday, a cell in the stomach called the mucus columnar cell. And again, I wanted to stress, don't worry about cell types unless I actually tell you to worry about them. And so it was called a mucus columnar cell. Now it's spelled M-U-C-O-U-S. So that's an adjective. It's a cell which secretes the substance, which is mucus. All right, so anyway, it's not the only cell that makes mucus. Lots of things make mucus, including a goblet cell. Mucus always looks the same in a microscope. It appears very light staining. And that light staining is because the molecular components, just there's nothing to stain. And so whether it's a mucus columnar cell in the stomach or a goblet cell or any other mucus secreting cell, it's going to tend to have a very light staining cytoplasm. But anyway, back to our discussion. We have ciliated cells, an occasional goblet cell. Here's the third cell type. It never makes it to the surface. This cell down here is called a basal cell. Basal cell. It represents the cycling stem cell for the tissue. So in other words, it divides by mitosis, and the daughter cells differentiate into perhaps a ciliated cell or a goblet cell, or the fourth cell type. So let's complete our picture here. Let me draw a couple of more ciliated cells just to show you that they are by far the most frequent. But occasionally, there's another cell type here. It reaches the surface, but it does not have cilia. In other words, I'm saying that the cytoplasm makes it to the surface, but it's not a goblet cell, and it doesn't have cilia, and this thing is called a brush cell. So brush cells probably have a sensory function. So as it turns out, pseudostratified is present in airways. And so there's these sort of local reflexes involving cough and sneeze and things like that, in which some particulate matter starts the thing off. And so a brush cell's probably involved in the sensory part of that. But anyway, let's stay with the big picture here. Pseudostratified has four cell types: ciliated cells, goblet cells, basal cells, brush cells. By far the most common cell type is the ciliated cell. Now there's lots of basal cells as well, but it's really the ones that reach the surface that catch our attention. Brush cells tend to be rare. You don't see them. And so let's identify these in the microscope first, and then we'll go from there. So let me switch over. All right, so first of all, what's going on here? You see this red line right here that goes right where the arrow is? Right here? That red line is the basement membrane. Now, many epithelia, in fact, the majority of epithelia, the basement membrane is really not discernible in the light microscope. For some reason, pseudostratified has an incredibly thick basement membrane, and so we can see it. So anyway, that's it. So that's the basement membrane. Ignore this junk out here, but this is the edge of the tissue. So again, body surface lined by an epithelium. And in fact, here, that's where the cells basically end. So these wispy things right here, those are cilia. So cilia extend out into the lumen. So, for example, this cell, that nucleus right here, is probably associated with this cell. So that's a ciliated cell, probably one right next to it. Here's some ciliated cells right here. So sometimes you get plane of section issues, so you get like a little peak of a little goblet, mucus droplet in a goblet cell. But here's a stretch of ciliated cells here. But now notice, this is a goblet cell. That's a goblet cell. Something I didn't mention, but notice the nucleus of a goblet cell is usually very dark, darker than ciliated cells. For some reason, mucus secreting cells tend to have very hyperchromatic, or that is, very dark nuclei. So that's a goblet cell nucleus, that's the goblet mucus droplet itself. And so goblet cells are fairly obvious once you know what they look like. So that's another goblet cell right here. Now meanwhile, what about these basal cells? Well, they're ones that the nucleus is going to be right down here on the basement membrane. So that's a basal cell, that's a basal cell. There's some more basal cells here. Okay, so that's 3 of the 4 cell types. So what about this 4th one? Well, the brush cell, remember I said it's relatively rare. You go the opposite direction. Sorry. So if we follow this along, we're going to see some areas in which the cells make it out to the surface, but they don't have cilia. So that's really the criteria. If it makes it to the surface like this, no cilia, call it one of those brush cells. Now, granted, that may not be the best example. Maybe I will go the opposite direction. So the key is I try to find a stretch in which I see a bunch of ciliated cells, and then, like right there, that's probably a good example. See where the tip of the arrow is? There's no cilia right there. So that cell is probably this cell called a brush cell. All right, now, back to our issue. Simple squamous, simple cuboidal, simple columnar, no problem. Pseudostratified, here's what you do. Identify it first as a tissue. And how do you do that? Well, it's going to be the only epithelium that has this configuration, this ensemble of cells, this combination of ciliated cells mainly, goblet cells, brush cells, basal cells. So no other epithelium has that combination. So if you see that combination, I don't care what it looks like as far as stratified or not, understand it's called pseudostratified, and it's by definition, head knowledge, is a simple epithelium. Again, think of the definition. Basal cells touch the basement membrane, but they don't make it to the lumen. So the definition does not include making it to the lumen. It includes that every cell contacts the basement membrane, whether it looks that way to you or not. Okay? So that's the last and definitely the most complicated of the simple epithelium. All right? Now, I don't want to get too much into this. You're probably aware of some of the functional aspects. And, I've been getting some questions. Do you have to know functional stuff for the test? And the answer is no, right? Correct? Yeah, that's a no. So, close your ears for the next 10 seconds. But what happens is there's a layer of mucus that sits right along this epithelial surface. It's called a mucus blanket, I guess. And what happens now is that mucus comes from a variety of sources. You're going to have a greater appreciation for the sources, at the end of our discussion today. But suffice it to say that goblet cells contribute a little bit to it. And so what happens now is that as air flows along the lumen, it's got stuff in it, like dust and pollen and particulate matter and other things, and that this material falls out of the flow of air and gets trapped in the mucus. So what happens is the cilia all beat in the same direction, moving this blanket along the surface. And the direction is towards the pharynx, because you basically swallow most of it. In some cases, you may cough it up. But for the most part, it's swallowed. Now, this whole concept, by the way, is called the mucociliary escalator. And it provides the ability to cleanse incoming air. Now, there's a bunch of other stuff we'll talk about when we do respiratory in the spring. But anyway, that's what pseudostratified is associated. Now, there are other epithelium that are called pseudostratified. I'll point them out as we go, but this is the sort of quintessential pseudostratified, airway pseudostratified. And again, find those cell types all together like that, and that's what you call it. Okay, so now, let's start talking about stratified epithelium, full-blown stratified epithelium. Now, just like there's 4 types of simple epithelia, there's 4 types of stratified epithelia. So, number 1, not every single cell contacts the basement membrane. That's what makes it stratified. So that's number 1, applying that definition. But number 2 is, remember, you name the epithelia based on the cell at the surface. Now this becomes important because the cells below the surface might look different. It doesn't matter what they look like. What matters is the cell shape at the surface. That's how it's named. So let's go through some examples. Here's the first one. Stratified. I'm going to do it backwards this time. Stratified. No, in fact, I'm doing it the same. Stratified squamous. All right. So, based on what I've just said, this is an epithelium in which not every cell contacts the basement membrane, okay? And number 2, the cells at the surface are going to appear squamous. Fine. It's a little more complicated than that, because as it turns out, there's 2 varieties of stratified squamous. One is called non-cornified. Now, that's kind of like an older term. So there's another term, which I prefer to use. I mean, you can use whatever one you want. But I prefer to call it incompletely keratinized. Okay? Now, keratin is a protein. And you'll understand why I've used this term incompletely keratinized in a moment, but let me go on. The other type is called cornified, also known as completely keratinized. But keep in mind that both are examples of stratified squamous. So let me show you the non-cornified variety first. Before I even write anything, let me just show it to you, and then we'll talk about it. Okay, so what you're looking at is actually, I'll just tell you, it's the pharynx. It's just, so I wanted to choose an organ that was covered by, stratified squamous, incompletely keratinized, and this is probably a good example. So this is the lumen right here. As I decrease the magnification, right, this is the lumen, and so this layer of tissue from here to here, that's an epithelium, and then there's a bunch of stuff back here, some of which we can already identify. I'm sure you can't see it at this magnitude. This happens to be skeletal muscle, and it goes on, et cetera, et cetera. But let's go back here to the epithelium for a moment. And now ask ourselves the question, even at this magnitude. The basement membrane's right here, and the surface cells are right here. Is there any way that these surface cells can contact this basement membrane? The answer is absolutely not. There's like 25, 30 cell layers in between that are going to prevent that. So, we're pretty comfortable calling that stratified. But now what's the other part of the equation? Well, we use the cell shape at the surface. And so what is that? Well, again, the basement membrane's pretty much here. Here's the cells at the surface. I'm going to give you three choices: columnar, cuboidal, or squamous. And of those, probably squamous is the best. They're certainly not rectangular. They don't look cuboidal, and so again, and first of all, what is the cell itself, the cell shape? Well, you have to use the nucleus to kind of figure that out. So a squamous nucleus is going to be sort of on the flat side, and so granted, not every one of these looks flat. So, something like that. That's a squamous looking nucleus. So this is stratified squamous. Now, I'm calling this incompletely keratinized. So why? So in the incompletely keratinized situation, here's some things to keep in mind. All right. Number one, the cells retain their nuclei out to the surface. Now that may sound like an odd statement, but it'll become meaningful for us in a moment. So the cells all the way out to the surface retain their nuclei. Number two, they tend to be called, that is the cells tend to be called keratinocytes, or keratinocytes, however you want to pronounce it, because they make keratin. Keratin is a protein. Now this epithelium, number three, lines internal body surface. So if you see stratified squamous incompletely keratinized, you know you're looking at an internal surface. The skin on the outside of the body is not incompletely keratinized, but it's the other one, completely keratinized. So the cells retain their nuclei, and they're called keratinocytes, and you know that it's lining internal body surface. Now, a couple more things. This epithelium is considered as a wear and tear epithelium, meaning that it's able to sustain, abrasion. So if you think about swallowing things, food particles could do a lot of damage to the epithelium. So the fact that it's multi-layered, these cells are pretty tough because they're filled with keratin. So it's usually considered as a wear and tear epithelium. And number five, it's often part of what's called a mucosa. I mentioned this yesterday, and I'm going to repeat it today. that the term mucosa refers to the epithelial lining of an organ in which it's covered with mucus. So mucus also has a protective function. So we're going to go back and look at this again with our sort of heightened awareness of what's going on. And that is, let's go back here to the same image. Let's drop our mag. You see these things down here? As it turns out, those are mucus-secreting glands. We'll study glands in more detail later. But anyway, and this is skeletal muscle down here. So these glands are secreting out onto the surface, and so this surface then is lined by a layer of mucus. Not that you see that. But anyway, this is what stratified squamous, incompletely keratinized looks like. And a key feature to keep in mind is that the cells out here at the surface, while squamous in shape, they retain their nuclei. Now, let me show you something, the next thing. Tell me what you think about this. Okay, here is the body surface right here. So that's the body surface. And here is the epithelium. It extends from here to here. Now, it's definitely stratified, wouldn't you agree? I mean, cells in here can't possibly, the base of the membrane is right along the surface. So it's stratified for sure, but now, what's the shape of the cell at the surface? Well, let's go out there. Take a look at this. And what we see is, it's kind of hard to even see cells, wouldn't you agree? I mean, here, yeah, those look like cells. But out here, much less cells. I mean, you might get an occasional thing that looks like a nucleus. And I suppose if we had to infer, we'd probably say squamous. But here's the point. The cells here at the surface lack nuclei. In fact, they lack organelles. Because what we're basically looking at now is we're looking at stratified squamous, completely keratinized. So here's some things to keep in mind. Number one, the cells at the surface lack nuclei as well as other organelles. So what are they? Well, they're simply cells which are filled with keratin and other things, and it really provides more of a water barrier. All right? Number two, there are distinct layers to this epithelium. I'm not going to describe them yet because we're going to have we're going to talk about skin in a little bit. Which brings me to three. It forms the epidermis of skin. So, stated differently, the only place in the body that you find completely keratinized stratified squamous is the skin surface, in which it forms the outer layer called the epidermis, which itself is subdivided into distinct layers, which I will describe for you shortly. And so, number four, there are distinct cell types. So in other words, instead of just calling everything a keratinocyte, that's not true here. So in other words, there's distinct cell types. So let's postpone this discussion until we do skin. But I do want to just go back to this image and show you what's going on here. And that is, drop our mag. So the epidermis now that we know this, goes from here to here. That epidermis is a tissue. Stratified squamous, completely keratin. It's called completely keratinized because the cells out here at the surface, which are continually sloughing off, those cells are only cells in name because they've lost their organelles. They're simply cell outlines, if it were, as it were, which are filled with keratin and other proteins, including a bunch of hydrophobic substances, which essentially, it's filled with lipid. So what the skin provides is a barrier for water-soluble things. That's a better way to put it. So anyway, we'll talk more about this, but I just want to stress it is an epithelium, but it forms this layer in skin called the epidermis. So that goes from here to here. Now, just to let you know, this is connective tissue down here. We're going to call that dermis. But again, let's forego most of this because we're going to, again, discuss all of this later. All right. Oh, actually, before I move on, let me just say this, just in case you're wondering. Not all skin looks like this. This happens to be a type of skin called thick skin, which is only present in certain places in the body, such as fingertips, palms, I guess, the palm of your hand, soles of your feet. Most of your body, though, consists of what's called thin skin. So let me show you that, just to show you that stratified squamous, completely keratinized can look a little different. And here it is here. Here's the body surface. The epithelium is basically going from here down to here. So you'll note there's definitely some layering going on. So this is called thin skin. This is much more typical. Okay? So that's the first of our stratified epithelia called stratified squamous. What's the next? Stratified cuboidal. Now, stratified cuboidal, let's just make sure we understand. It's going to be an epithelium in which the cells at the surface cannot possibly touch the basal membrane. Okay, that makes it stratified. And those cells at the surface have the shape of being cuboidal. Now, as it turns out, stratified cuboidal is an extremely rare tissue type in the body. In fact, it's only found, as far as I can think at the moment, it's only found in one place. And that is the ducts of eccrine sweat gland. All right, what's an eccrine sweat gland? Well, instead of me telling you all about that, because again, we're going to talk about skin later, suffice it to say there's different types of sweat glands. The most common one is called eccrine glands. And basically what happens is this. If this is the epidermis, and this is the dermis, you're going to find out in a second, this part down here is called the hypodermis. And down here is where we'll talk about what's called the secretory part, or secretory portion of an eccrine sweat gland. And it's got a duct that extends its way all the way to the surface. So this is the duct. So the point is that an eccrine sweat gland has two portions, a secretory part and the duct. We want to focus on the duct because that is what has stratified cuboidal. So let me show it to you first, and then we'll talk about it. All right, so this is where we are, is I didn't change slides. So this is this thin skin slide we just looked at. And so I'm down here at sort of the dermal-hypodermal junction. And I'm finding an area here which has eccrine sweat glands. But you'll notice there seems to be two kinds of things going on. So number one, you have this sort of pale staining stuff like this. Well, that's the secretory portion. That's where sweat is made. So this is the secretory part right here. I'll fill you in on the details later. But what we want to focus on these guys right here. That is a duct of an eccrine sweat gland. So we want to focus, though, of course, on the tissue. So what are we looking at? Well, we're basically looking at, here's the lumen, put an L for lumen. The cells which line the surface basically look like this, so more or less cuboidal. And then there's another layer, and there's kind of like two layers. And so not that you see this, but I'll just kind of draw a line in here. We have one layer and then two layers. So stratified cuboidal then tends to have two layers, but the inner layer is certainly cuboidal. So as a name, stratified cuboidal is extremely fitting. But I just want to make mention of another thing. There's something called the cuticular border. And this is a very acidophilic structure. So let me show you what I'm talking about. So this is the duct. And now, just bear with me for a second. We'll lose this. All right, notice, just focus on one of these things. All right, lumen is the space in the middle, and here's this inner layer that I was talking about. See how those cells are basically cuboidal? The cells on the outside are sort of cuboidal, maybe a little less so, but it doesn't matter, because the name is based on the cells that border the lumen, in this case cuboidal. So, term, stratified cuboidal, and this happens to be a duct of an eccrine sweat gland. Now, the secretory part is actually a simple epithelium. Notice it's only one cell layer thick. But again, we'll come back and talk more about eccrine sweat glands later, but let's stick with the tissue types. So there's the only place I can think of where stratified cuboidal is found. All right, so number 3, stratified columnar. That has an even, well, I shouldn't say even, it also has a limited distribution. So remember what we're talking about here. We're talking about more than one cell layer, but the cells bordering the lumen are columnar. Honestly, the only place that I have any material for, and the only place I can think of where it's actually, well characterized as being stratified columnar is the penile urethra. Probably didn't expect that today, but anyway, we're going to show you the penile urethra right now. All right. So, if you remember your gross anatomy, you'll remember that within the shaft of the penis, there's erectile tissue. And there's different parts. There's what's called the corpus cavernosum. They're actually paired, corpora cavernosa, kind of right and left together. And so this is what you're looking at right here. So it's actually, I guess this is relevant to our discussion. If you look at the lumen, you'll see that it's lined by a simple squamous epithelium. That's interesting, because these are actually blood spaces. So it's obviously not filled with blood at the moment, but it would be if this male had an erection. And so this lining then is actually an endothelium. Okay? So that's what erectile tissue looks like. You know, again, you will consider this in greater detail later in your education. But what we want to do, though, is go to the other area of erectile tissue, which is the corpus spongiosum, because what we want to look at is this thing right here. Because this is the penile urethra. So, to sort of use our rules of classification, notice that here's the lumen. The epithelium is basically from here to here. More. So, doing this again, a good area. So it's from here to here. So a cell out here can't possibly touch the basal membrane because there's too many cells in the way. The cells down here, you know, their shape is, you know, probably more like cuboidal. But remember, the naming sequence, though, is based on the cells that line the surface. So if you look at these nuclei right here, can you envision that each of these is like a sort of rectangle? Now notice the long axis of the rectangle is perpendicular to the basement membrane, so that's important. But it's definitely stratified. Surface cells are columnar, hence the term stratified columnar. Now here's the thing, it's not found, like I said, in very many places in the body. So literally, an image like this, it almost would be fair to say, what organ is this? Because you would recognize it as an epithelium, specifically a stratified columnar, recognizing it's only found in the penile urethra. There you go. So sometimes that's the way these decisions are made. So going back to our drawing board, as it were, basically what we're looking at is this is the basement membrane. There's a bunch of cells down here, but the cells lining the surface basically are columnar. So again, it doesn't matter what the cells look like down here, it's the cells at the surface. Now, remember we said there's 4 stratified epithelia? So the fourth one is kind of like the quote-unquote specialized one. It's called transitional. Now, transitional epithelia, the transitional epithelium, the name is a little unfortunate because they named it before they really understood what they were talking about. Because they thought that the epithelium literally transformed itself based on the conditions of the organ. So it turns out that is not what happens, but the name stuck, and so our job is to identify it. And so here's the thing, it's only found in a certain number of places as well, and they pertain to the urinary system. So things like, I'll work backwards, the bladder, the ureter, a major calyx in the kidney, a minor calyx. You may not know what these things mean, but don't worry. I'll show you, I think I'll just use the ureter as an example and show you what we're talking about. So basement membrane, couple layers of cells. What makes this epithelium identifiable is the cells that line the surface. They do this. They're rectangular, but notice the long axis of the rectangle is parallel to the base of the membrane, not perpendicular. Every other example that we've talked about of columnar has been perpendicular to the base of the membrane. This is now the exception, it's parallel. These cells are oftentimes called half cells. Nucleus, nucleus, nucleus. And so what you're looking for is this type of appearance. Now, transitional epithelia are known for their extremely low transepithelial permeability, meaning that solutes have a hard time crossing, and namely water as well, this epithelium. And if you think about kind of what's happening here, urine, which is contained within the lumen of these spaces, can get pretty hypertonic. depending on body conditions. And so if you're trying to conserve water by excreting concentrated urine, what you don't want to do is have water flow by osmosis from below the epithelium, so they're very watertight, very low water permeability. So let's just say low water permeability. Now, again, we're going to study these organs, later in your education, but let me show you what the epithelium itself looks like. Okay, so this is the ureter. Not a very big organ. This is one side of it. This is low magnification. This is the other side. That's the lumen. The lumen is kind of thrown into this kind of star-shaped appearance because this is smooth muscle which contracts upon exposure to these fixatives. But what we want to do is we want to identify what this epithelium looks like. So let's do that. If we come in here, and look around a little bit, find a good area. All right. If that's the lumen, and this is the basement membrane right here, obviously it's stratified, right? These cells cannot touch the basement membrane. But notice how they're kind of rectangular. Like that one is, this one is, these guys right here. So the fact that the long axis of the rectangle is parallel to the basement membrane makes this not stratified columnar, which is, you might be tempted to say, but instead it's the specialized one called transitional. Okay? So those are the four stratified epithelium. So let's just pause for a minute and just think about what we've said thus far. So, number one, when we talk about epithelia, the first thing to recognize, and this is something that will become important when we do a review next Monday, and that is, if it's a body surface, you should expect it to be covered by an epithelium. That's step one. Step two is determine, is it stratified or is it simple? Again, keeping pseudo-stratified kind of to the side for a moment. And then, if it's stratified, then it's either one of these four. If it's simple, it's simple squamous, simple cuboidal, simple stratified. Okay? So the basic identification of the tissue, again, with a little practice, I think you'll come to find it's not so bad. But there's some additional things that we need to talk about under this discussion of epithelium, such as the next topic, which is surface specialization. Remember we said that a basement membrane imparts polarity to epithelial cells. And as a result, you can distinguish between what's called an apical membrane versus the basolateral. Well, the apical membrane is specialized based on the proteins that are there. Certain ion channels, certain transporters become specialized in apical versus basolateral. But the other thing is that the apical membrane can be thrown into a number of surface specializations, for which I am going to describe three for you. Here's the first ones. They're called microvilli. If this is an epithelial cell, what you can see oftentimes, or what you find oftentimes, better word, is the apical membrane is thrown into a series of finger-like projections. So this would be the next cell over, etc. Now, each of these is called a microvillus. Microvilli is simply plural. But here's a very important observation. Microvilli are, for the most part, below the resolution of the light microscope. In other words, cannot resolve individual microvilli. So what that means then is that, well, if you can't see individual microvilli, can you see them at all? And the answer is yes. You can see, we'll say, composite indicators. So what does that mean? Well, here's the first example. It's called the striated border. And this is what you find in cells called columnar absorptive cells of the small intestine, and actually large intestine, but we're going to look at the small intestine. Let me show you what we're talking about. Now this hopefully will look familiar. We looked at this the other day. That's the way that is. So, you remember this image? And that is, I was using this to illustrate the epithelium simple columna. This happens to be the ileum. And if we look out here, I'll get this in focus, you'll notice, interesting, we see goblet cells, don't we? All right, so goblet cells, as I think I warned you, are not just present in pseudostratified, they're here as well. So in the lining of the small intestine and large intestine, for that matter as well, there are goblet cells, but they're not the only cell type. These cells right here, which are non-goblet cells, are called columnar absorptive cells. And that's a good name for a reason. Because if you look right here, so these are goblet cells, but this columnar absorptive cells, here's a stretch of them, you'll notice that they have a kind of line, red line along their surface. And that line is called a striated border. That line is created by microvilli. In fact, it's more than microvilli. It's the microvilli plus the whole junctional complex and everything. But anyway, in the small intestine, you call that a striated border, and it's, the name's a pretty good one because it kind of makes it look like it's striated. Okay? Now, using a different type of preparation, I want to show you something else. Same organ, but now this is stained sorry, I didn't even leave you hanging with that sentence. I'm just looking for a good area here. So this same organ, this is the ileum, but it's stained in such a way, it's called PAS stain, and it's stained preferentially mucus stuff, and so you see how the goblet cells really are clearly indicated. But what I want to show you, though, is if you look in between the goblet cells, like right here, those are columnar absorptive cells, and the striated border tends to be stained as well. Because, as it turns out, there's this concept known as a glycocalyx. Have you ever heard that word before? Glycocalyx, it's a term that describes the fact that there's a bunch of different molecular components in and around all these microvilli. And so what this illustrates is that there's actually, mucopolysaccharides and things like that that are present here. But anyway, the thing is called a striated border, and I just thought that looking at this, it would clearly indicate where the striated border is. So, surface specialization, microvilli. Unfortunately, that's not the only example of microvilli. And I say unfortunately because there's another example. I don't know why, but they couldn't call it the same thing. They called it a brush border. And these are in proximal tubule cells of the kidney. Now, I'm not going to sit here and tell you all about the structure of the kidney at this point, but I'm simply going to show you what a brushed border looks like, and then we'll sort of make a comment about this. So I have to go to the appropriate area of the kidney, which I've just done. And as soon as I find it, I'll switch over and show it to you. Okay, so as it turns out, this thing right here, that's the lumen of a structure that you will learn later as a proximal tube. This is something called a distal tubule. And if you notice, the luminal surface of these cells is pretty smooth. As in contrast to the luminal surface of these cells, which look a little irregular. The distal tubule is lined with cells that don't have microvilli. Hence, we say they lack a brush border. The proximal tubule, of which this is another one, key feature, I suppose, is it's rather indistinct where the cell ends and the lumen starts, but cells of the proximal tubule also are lined by microvilli. And instead of a striated border, it's called a brush border. This looks more like a brush to these people than a striated border. But anyway, it's the same thing. That is, they're composed of microvilli, but you use these two different names depending on what you're looking at. Now, I can feel the tension rising, so don't worry so much about it. You're not going to be tested on this at this point, but simply realize that microvilli, because you'd have to know, oh, am I in the small intestine? Oh, am I in the kidney? So you'd have to know this before you could really make that identification, and I don't think we're going to hold you to that at this point. That will come later. But anyway, that's the first of three surface specializations. So I'm going to show you the other two, then we'll take a break. The next one is something you've seen already, and that's cilia. Now, cilia can be seen. And that's the difference because they're longer than microvilli. Now, cilia are found in many places in the body. But I'm going to go back to this pseudostratified just because we were just there. So let's do that. But pay attention now to the cilia. So let me just find an area. Okay, so here it is. So if I focus up and down, you can kind of almost see these wispy things. And those are the individual cilia. You can certainly see the length of them, right? They go from here to here, so they certainly they can be seen more clearly, let's say, the microvilli. Instead of presenting as a line, they present as, more than that. So anyway, cilia, again, another surface specialization. Unlike microvilli, as you're probably aware, cilia are motile, they move. They're composed of microtubules instead of other proteins for microvilli, so microtubules. are found in cilia. And so, key difference between microvilli and cilia really has to do with whether you can see them or not. You see an indication of microvilli, but you can see individual cilia. Now, the last of the three surface specializations are shown here. Again, I just want to focus first. Okay. So, they're called stereocilia. Also not present in many places. We're going to look at them in the epididymis. So, again, avoiding a lot of the detail. What you're looking at is the lumen of the epididymis. That's where the arrow is. The cells that make up this epithelium, it varies from a simple columnar to sometimes people call that pseudo-stratified. Don't concern yourself with that. But what you need to understand, though, is you see these wispy things right here that extend out into the lumen? Those things, those wispy little things right here are called stereocilia. They're larger than cilia. They're not as uniform in, length and stuff. Also, importantly, they're non-motile. They do not move. And so they got named stereocilia for some reason. They provide little microenvironments. If you remember, the epididymis is where spermatozoa finish maturation. So anyway, it's the third surface specialization. So essentially, you got microvilli, cilia, stereocilia. All right, so let's take 5. We come back. We're going to talk about exocrine glands because they have a relationship to epithelium.